13th question. Crossing of an individual with double homozygous, that is recessive, double recessive plant. Double recessive plant, that is, suppose small t, small t, small v, small v. In order to determine its genotype, that is, it is homozygous or heterozygous, homozygous or heterozygous for a character. In that case, better to perform test cross. By the test crossing, we can know whether this plant carries gene for homozygosity or not. Capital T, small t, capital V, small v, this is the tall plant with violet flower. If it is crossed with the small t, small v, small v, then if such plant is obtained, if this plant is obtained as a result, then hence confirmed this particular hybrid plant is homozygous, F1 plant is homozygous or heterozygous. This can easily be known by the test crossing. It can easily be known by the test crossing. Crossing of organism with the double recessive, sure. That is, that particular organism. If suppose this wasn't hybrid, that is capital T, capital V, in that case this will not be obtained. Hence prove that plant is pure. And if it is hybrid, then this result will be there. By the test crossing, genotype of the hybrid can easily be known. So test for this, test cross is done. And what is test cross? That is cross between F1 plant and the recessive parent. F1 plant with the recessive parent is known as test cross. It is a back cross, but specifically if recessive parent is taken, that is test cross. To know the genotype of F1, so best answer is test cross. A dihybrid cross, the F1 heterozygous plant are self-fertilized. What is F1 heterozygous? That is capital T, small t and capital V, small v. That is plant is tall but hybrid tall. Plant is tall, that is hybrid tall. If it is selfing is done, I mean to say same genotype, or we can say male and female gametes are from same source. Male and female gametes are obtained from the same source. If the cross is made, then you know the phenotypic ratio is this one. That is dihybrid plant on selfing produces four types of phenotype in unequal proportion, and there is a formula which I have already described in a regular session, that is apply the formula 2 raised to power n to know the phenotype in F1 generation. This formula is applied to know the types of phenotype in F2 generation, not in F1, F2 generation. And this cross is dihybrid cross, then the value of n is 2, that is 4 types of phenotypes are obtained. But in what proportion? That very simple, that is in monohybrid cross the ratio is 3:1. In dihybrid cross you should multiply 3:1 with the 3:1. Then check this one, 3 3 ja 9. 3 1 ja 3, 1 3 ja 3 and 1 1 ja 1. That is, this is the answer. That is 2 monohybrid cross 2 monohybrid plants. If you multiply that ratio, you will get easily without making checkboard. Monohybrid ratio is 3:1. If plant is dihybrid, then better multiply 2 monohybrid ratio. You will get the answer in very brief. Without Punnett square, without any short of word. So this is dihybrid ratio on the basis of phenotype. This is the dihybrid ratio on the basis of phenotype. That is phenotypic ratio in dihybrid cross. This is phenotypic ratio of dihybrid cross, generation F2. Dihybrid cross. Cross is dihybrid and ratio is on the basis of phenotype. The traditional way, if you want to make the checkboard, it is also possible, but it will takes time. It will take time. How? Take a normal plant, that is capital T and capital V and small t, small v, small v. This is the parental generation. Then see the F1 plant, hybrid for both the traits. This is F1 plant. Then selfing is done, so you will get the F2 generation. In that case, you have to make the entire checkboard comprises of 16 board and analyze each one. So better, that is, this is monohybrid area, this is monohybrid area. If this particular plant is selfed, you will get the 3:1 ratio. Similarly, this is also monohybrid. So if it is crossed, then multiply with 3:1 is to 3:1. Then you will get easily this answer without making checkboard. Then a cross between pure tall, that is capital T and green seed. Green seed is recessive. The green color of a seed is recessive. That is small y is crossed with the pure dwarf, that is small t and that is this one. Then the number of phenotypes produced, very simple. Take the gamete of first one and take the gamete of the second one, apply capital T, capital Y, small y. This is first generation, and if the question is asked from this particular generation, then answer is 1. All the plants are of same genotype and same phenotype at this level. All the plants are with same genotype and the same phenotype. Plant is pure tall, that is capital T, but seeds are green and green color is recessive. If it is crossed with the, that is I am solving again, if it is crossed with the pure dwarf plant, dwarf plant is already pure, no need to write pure with the dwarf plant. It is a recessive feature that is always pure. That is small t, but for one feature is dominant and pure, yellow color. Then pure dwarf and the green and the what is the yellow seed is crossed with the that is only one type of phenotype, that is capital T, small t, capital Y, small y. All the plants are of this particular type. All the plants are of this particular type, because take the gamete of this particular plant and take the gamete of this particular plant, you will get only one plant, one type of plant, that is they all have this genotype. Hence answer is 1. The answer is 1. In a cross between the individual genotype, that is the genotype of a plant is already given, this is dihybrid plant. See, this plant is dihybrid. Yes, resulting the number of offspring, yes, 16 offsprings are obtained in what? That is in F2 generation. Number of genotypes of this particular plant, capital T, small t, capital R, small r. And second option is capital T, small t, capital R by the selfing of this plant. How many are there? Apply the monohybrid ratio. Apply the monohybrid ratio directly without making checkboard. In monohybrid cross, this is 2/4 and the second one is also 2/4. That is, dihybrid plants are obtained in 4/16. And what about this one? 2/4 into 1/4. Apply bilkul monohybrid cross ratio apply, then answer you will get 2/16. 2/16. Then answer is 4 and 2. Answer is 4. That is, these plants in 4 and these are in 2. Otherwise, make the entire checkboard and R apply this one. That is, if this plant is selfed, then actually I apply the monohybrid cross. If this plant is crossed, you will get T 1/4, T small t 2/4 and small t 1/4. That is by this one, and if this is selfed, same ratio is obtained, and I multiplied the value, I apply the value and directly try to find out the answer and get it. If you want to know once again, then the idea is very simple. Write capital T, small t, capital R, small r. This plant is dihybrid plant and its selfing is done. That is male gamete and female gametes from the same source. Then I have separated these two. That is capital T, small t, this plant is monohybrid. If it is selfed, then you will get this checkboard. Capital T, capital T, capital T, small t, capital T, small t, small t, small t. In which proportion? 1/4, 2/4 and 1/4. And I did the same thing with this one. That is, selfing of this particular plant is done. Then apply these values. Actually, these values are important to know the phenotype, know the genotype without making checkboard. And apply this value over this one, because the number of plants, the probability of appearance of dihybrid was asked. How many plants will be there in F2 generation? So kept the value of 2/4 and multiplied with the 2/4. That is in the same ratio these two are obtained. So better answer 4 by 16. And for second one, capital T, small t, capital R, write the value 2 by 4 here and 1 by 4 here. You will get the answer 2 by 16. So answer is 4 and 2. 4 and 2. This is my answer. And question number 16 is solved. Question 17. Number of male gametes having both dominant gene YY and Y, both dominant gene. First of all, this genotype is given and how many types of gamete it will produce. That is capital Y, capital R, small r. Then first possibility, Y with capital R, YR. Then Y with a small r, capital Y, small r. Then see this one. Same gametes are obtained and same obtained. That is only two types of gamete is produced by this plant. Two types of gametes are produced by this plant. So if 1000 is given, then any one probability of both dominant, that is capital Y and capital R, that is they are obtained in 1 by 2 manner. So divide this one 1 by 2, you will get the answer 500. This is the correct answer. Number of male gametes having both dominant. Number of male gametes, that is having both the dominant. Recheck this one. Recheck. Write the genotype first. Capital Y, capital R, small r. This plant is actually monohybrid plant and it produces two types of gamete. This plant is actually monohybrid plant and produces two types of gamete. Because if you apply the shortcut 2 raised to power n, then monohybrid plant is n value of n is 1. Then your answer is two types. Then let's see capital Y, if it is in this combination, then first gamete is capital Y, capital R. That is both dominant genes are there with the gamete. And the second one, capital Y with a small r, and no other possibility is there. That is, this type of gamete is produced in 1 by 2 and this type is in 1 by 2. So how many out of 1000 plant? Then directly multiply this one. Find out the ratio. Answer is 500. That is 50% of 1000 is 500. 18th question. A self-fertilizing trihybrid plant. Trihybrid plant, that is capital A, small a, capital B, small b, capital C, small b. This plant is trihybrid. There is a shortcut to know the types of phenotype and types of genotype in F2 generation. Formula of phenotype 2 raised to power n. Formula of genotype is 3 raised to power n. And this cross is trihybrid. That's why the value of n is 3. 2 raised to power 3 for phenotype and 3 raised to power 3 for genotype. That is 8 types of phenotype and 27 types of genotype. For the checkboard related question, this is checkboard related question. If we take one pair, that is capital T, small t, then you will see this checkboard is obtained containing 4 different types of zygotes. 4 types of zygotes. If we apply this formula, if we use the dihybrid plant, in that case, that is 16 containing board will be obtained. If we take trihybrid plant, in that case, first of all types of gamete apply this formula 2 raised to power n. You will get the answer 8. Then 8 types of gamete, but 8 types of gamete, 8 here in this line and 8 types in this line, you will get the answer, that is 8 into 8, that is 64. So answer number 1 is correct. Answer number 1 is correct for the second generation. Answer number 1 is correct is for the second generation. How? Let's repeat again. Capital A, small a monohybrid. If we write capital A, B, small b, then 2 hybrid for 2 feature, dihybrid plant, and capital C, small c, that is trihybrid plant. How many types of gamete produced? There is a shortcut, that is apply this formula 2 raised to power n. N indicates hybrid. This plant is trihybrid, that's why the value of n is 3 and answer you will find 8. The 8 types of gamete. First of all, gamete type is 8. If we write 8 male gamete in this line and 8 female gamete different types in this line, you will find 8 8s are 64 types of zygotes. 64 zygotes you will get it. This is the answer of 18th question. 8 gametes in this line and 8 gamete in this line. Better to multiply them. 8 into 8, that is 64. 19th question. Dihybrid cross explains. Dihybrid cross explains, that is exclusive explanation from dihybrid cross. Then this answer is correct. That is, independent assortment can be studied only by dihybrid cross. Independent assortment can be studied by dihybrid cross. Whereas from monohybrid cross, we can study dominance at F1 level and segregation at F2 level or by test cross. From monohybrid cross, 2 laws can easily be studied. Number 1, dominance. Number 1, dominance. And number 2 is segregation. Number 2 is segregation. These 2 laws can easily be studied by monohybrid. But if you want to study independent assortment, then minimum cross required is dihybrid. Minimum cross required is dihybrid. Mendel's first law is known as simply law of dominance. Law of dominance according to NCERT. Law of dominance according to NCERT. Which of the following Mendel's law of segregation? Genetic material equally distributed during meiosis or mitosis. Wrong. Then each gamete receives 1 gene for 1 allele. This is right. Genetic material enters only in 1 gamete. This is wrong. That's why all of these is not possible. That is, B is correct. B is correct. Mendelian recombination. Simple answer, that is due to independent assortment. That is due to independent assortment. This plant is hybrid tall and for flower color, that is violet flowers are there, but it is for hybrid. But when we made the cross, our selfing is done. You will see 4 phenotypes: tall and violet, tall and white flowers. This is new combination. Then dwarf with violet. This is again new combination. And dwarf with white. That is 2 different types of combinations are seen, and this is because of independent assortment. And the reason behind is, that is 2 or more Mendelian factor if they are located on 2 different chromosomes. I mean to say height and flower color, if it is not on the same chromosome, then their assortment is free from one another. It is not dependent on one another. That is known as independent assortment. That is two features. I mean in this case height and flower color. If their factors are not on one chromosome, I mean they are not on the same chromosome. At one chromosome, capital T is there. On the same homologous chromosome, identical position, small t is there. This is the pair of homologous chromosome. Similarly, with respect to the color of flower, capital V and small v. I mean to say that is height and flower color. Height and flower color, two different types of characters. If their gene or allele located on two different chromosomes, then their inheritance, that is in next generation, is not dependent on one another. Suppose if they are on one chromosome, I mean flower color and height. This is height and this is flower color. In that case, they will not assort independently because they are on same chromosome, and this is called linkage. So, Mendelian recombination is applicable for independent assortment. Mendel was not able to say anything about the recombination and crossing over. Mendel was wise actually. He didn't study those characters which are linked. That is better one. Trait he selected were not linked. This is the correct. If they were linked, then he was not able to give the law of independent assortment. So that's why this answer is right. He didn't study linked features. Phenotype of an organism is result of, that is genotype and environmental interaction. Genotype and environmental interaction. For example, see this one. If this genetic makeup is there, we say plant is tall. If this genetic makeup is there, then we say it is dwarf plant. Sorry, tall plant. Again tall but hybrid tall. But if the genotype is in this manner, then we can say plant is dwarf. So definitely genotype play an important role in determining the phenotype as well as external condition. Environmental interaction is also responsible because it causes mutation also. Which of the following represent the deviation of Mendelian concept? That is independent incomplete dominance. In Mendelism, it is applicable for the pure behavior of these two genes. If there is a change in any other inheritance, then definitely Mendelism will be affected. So, incomplete dominance is deviation of Mendelian dominance. Similarly, if linkage is there, if we study linkage, then it will be the deviation of independent assortment. But here only one is given. So, that's my answer, that is incomplete dominance. Incomplete dominance is the deviation of Mendelian dominance in this case. Others are right. Segregation studied by Mendel, and it is a universally applicable principle. Segregation is universally applied principle. Universally applied. This law is universally applied. Universally applicable law. But for others, no. This is minimum cross required dihybrid. Purity of gamete is also called segregation. This is also called segregation. This is also called segregation. Segregate, that is separation. Separation of Mendelian factor, it takes place during meiosis, and meiosis takes place generally at the time of gamete formation. So the Mendelian factors get separated. Thank you.